me in this?
Yes,
Yes,
absolutely.
okay. So I guess I'll stand on this side then. Let's see. I was a proud son to, no, I'm just kidding. Anyway, I got my bachelor's degree at Ohio State University, went on to, in biology, went on to do a master's degree in science at the University of Arkansas, Fayetteville, where I actually discovered adult-derived stem cells and the material that controls how they work. I went to Texas Tech University in Lubbock to pick up a PhD in anatomy. And I learned histochemistry at that point, which is biochemistry on tissue sections, to get a better feel for what the cells look like and the glycoproteins and proteoglycans interfaced with them. Then, because being an anatomist, you could only what I would call slice and dice, it's a little cutesy name for it. So I went on and did a postdoc in biochemistry, which cutesy name would be grind and fine, at Case Western in Cleveland to be able to isolate the compounds so I could apply them back on the cells to see how the cells would work. And then I was wondering, is there different ways that we could isolate the cells? And at that time, antibodies came into being. So I took a job at Rush Presbyterian in Chicago and ran their hybridoma facility for a while, learning how to generate antibodies and things like that, and found out that was not the way to go. It was like a shotgun approach, too much of a shotgun approach. Then I went down to Macon. I've been there now for 21 years. Went up through the ranks from assistant professor. I'm now full professor there. And I teach anatomy to our medical students. I teach research to our pediatric residents and to our obstetrics and gynecology residents, and anybody else that I can drag into my laboratory. So if you combine, you're too close, you might be involved in my research, whether you like it or not. But anyway, moving along. So I have a biasness towards adult stem cells. I've been working in this research field for basically, now it's over 35 years. And it started when stem cells weren't even recognized to exist, especially adult stem cells. As originally you were taught that the cell that you were born with is the cell that you die with, which to me didn't make any sense anyway, but that's what we were being taught. And I was only in the late 60s that they began to start seeing that, well, that wasn't exactly correct. There were immediate precursors so the cells could wear out and be replaced. And then we went on from there. A lot of the procedures we developed on their own because there weren't kits that you could take off the shelf and do things. And the procedures were developed empirically based on cell-cell interactions. And what I'd like to do tonight is compare and contrast embryonic stem cells, which a lot of people, if you say, I work with stem cells, they go, ah, embryonic, and they have a heart attack. No, I work with adult stem cells, but before I can get the word adult out, they're already on the floor gasping for breath. So what I want to do is tell you about both types and let you figure out which one you think is best. I'll just leave it at that. But I'm going to try to be as non-biased as I can. But the whole idea is, why is everybody so interested in stem cells? Anybody know? Okay, what do you think?
They're precursors to other cells in your body, like nerve cells, blood cells.
Okay. Why is that so important, though?
Because you can start out with a stem cell and you can trigger it so that it can become
Exactly. But still, why is that important?
because it can solve diseases such as muscular dystrophy.
But why is that important?
And I don't know, I'm stuck now.
Because nobody's willing to donate the organs. That's why it's important.
Oh. Okay.
That's one of the reasons. Anyway, you have end-stage organ failure and tissue loss. U.S. healthcare costs, this slide was generated about 5 years ago. At that time, 80+ million surgical procedures a year, 40 to 90+ million hospital days, and 5 years ago it was $400 billion a year, just because of that. And treatment options, you could have transplantations, you could have surgical interventions, and right there's the key. Whoa, what is this now? Okay, I don't touch the smartboard. That's what I do. Okay, I use the pointer. Okay. The whole point is critical donor shortages. How many people in this room, on their license plate or on their driver's license, says that they will donate their organs? Not bad. Pretty good, actually. Most people don't, this sort of thing. Donor shortages is the big thing. There's not enough people walking around that are willing to donate their organs for other people. And they also have issues with long-term morbidity and long-term mortality issues as well. So, if donated organs are not available in sufficient quantities, can tissue be grown for transplants? And that's the whole idea of stem cells. Now, if you use a stem cell, you want a cell that's easily accessible, so you don't have to go digging for it all the time, so to speak. It has to have unlimited proliferation potential, meaning take one cell and it would fill this building with cells. All you need to do is go after it once. Would form any cell type in the body. So you don't have to go for different types of stem cells. And to paraphrase Monty Python here, that is the holy grail for stem cells. Now, everybody has their different definitions for stem cells. I have mine, so we'll go through mine so we all start on the same page. A totipotent stem cell is a stem cell that will form the embryo, the placenta, and the gametes, which are the sperm and ova. So it forms everything. A pluripotent stem cell, on the other hand, has lost the ability to form the placenta and lost the ability to form the gametes, but will still form embryonic structures. Then you have what are called germ layer lineage stem cells. And when each of us develop, we split into 3 germ layer lineages. We split into ectoderm, which forms our skin, our central nervous system, hair, nails, things like that, teeth. Our endoderm, which lines the inside of the body. You're kind of like a hot dog, so to speak, an outside and hollow tube inside. So it's the lining of your respiratory system, the lining of your gastrointestinal system, things along those lines. And then what I like to call the packing tissue in between, which is the mesodermal stem cells. So they form the cartilage, the fat, which a lot of us have, or love handles, or whatever you want to call it, bone, connective tissue, scar tissue, blood vessels, blood cells, things along those lines. So each of us have those. Then we have what are called progenitor cells. Now these cells are already committed to a particular cell type. You can have ones like the multipotent hematopoietic stem cells, and there are 35 different cell types within the hematopoietic lineage. You've got white blood cells, you've got red blood cells, you've got dendritic cells, you've got Langerhans cells, antigen presenting cells, yada, yada, yada. But there are about 35 of them. These cells will form anything of that particular lineage, but they won't form anything outside that lineage. They won't form neurons, say, no matter what you read in the newspaper. You have tripotent stem cells, which will form like 3 cell types, bipotents, which will form 2 cell types, and unipotent progenitor cells, which will form 1 cell type. Like a myoblast will only form muscle, fibroblast will only form connective tissue, and my big worry here is a diploblast, which will only form fat tissue. Okay, so, but we can actually separate this into 2 major categories. One category is stem cells, and the other category are progenitor cells. And the way we separate these 2 categories is stem cells are plastic. They can form any downstream cell type, so a totipotent can form a pluripotent can form germ layer can form progenitor, can't go up, it can only go down. And at least these things that are called stem cells here have unlimited proliferation potential. I have a clone of cells that is out from human, that is out 690 cell doublings, it's out probably over 700 by now. That's 2 to the 690th power, which means 1 cell could fill this whole building, this sort of thing. Now we get to progenitor cells. They are not plastic. They cannot form a whole bunch of different phenotypes. They're locked into a particular cell type, and they have their own biological clock. And for humans, that's 50 to 70 population doublings. And don't get too scared, that's about 120 years of life. So, it's not bad unless you buy a truck or something. But anyway, if everything goes and you're healthy, you can live to about 120 normally. That's about a 70 population doublings. But then they are programmed to senesce and die. That's their normal programming. Okay? So those are progenitor cells, stem cells, unlimited. They just keep on going, doing their own thing until they're committed to become progenitor cells. Then they assume everything that a progenitor cell is. Now, how do we derive embryonic stem cells? Well, how do we get an embryonic stem cell to begin with? Well, we have fusion from mom and dad. Ova and sperm forms a cell called the zygote. that cell will split into 2 cells, and each one of these cells are called blastomeres. And these blastomeres are totipotent. They'll form everything: placenta, germ cells, and embryo. And these things will go from 2 cells to 4 cells to 8 cells, basically to 16 cells in a morula, a solid ball of cells. And then they'll form what's called a blastocyst stage. And the blastocyst stage, traditionally we teach, is the first differentiation step. It's actually not, it's the second one, but for you guys it's the first one. Where we have a separation of what will form the embryo, which is the inner cell mass, to what will form the extraembryonic membranes of the placenta, called the trophoblast. So that's the first differentiation step. So this is called the blastocyst stage. Now at this stage, what they discovered was if they disrupted the trophoblast cells, pulled out this inner cell mass, disrupted them, and put them in culture on a feeder layer or with inhibitors, they could proliferate these cells, grow them up to mass quantities, as long as they were in the presence of an inhibitor, because these cells are programmed to form embryonic tissues and will do so without the presence of an inhibitor. Automatically, they'll just start and do it. But if you have an inhibitor present, it stops that process and allows them to proliferate. So you can do ad nauseum, get as many cells as you want. Then when you have the number that you need, you pull away the inhibitors, and they form these entities called embryonic bodies. And then these embryonic bodies will spontaneously differentiate into the cells of the embryo. Now normally they do it so you come out with an embryo. If you do it in culture dish, they form the cells of the embryo, but it's all just one big mass of stuff. But they call this a pluripotent stem cell because it won't form the trophoblast and it won't form gametes. Okay? Now, a group in Japan about 4 years ago now said, what if we back up one stage and go after blastomeres instead? Since they are totipotent, can we do the same thing that they did on the inner cell mass cells? So they take blastomeres, which will form everything, at the morula stage, take them out, put them in culture, as before, in the presence of inhibitors, allow them to proliferate, increase in mass, and when they're ready, pull away the inhibitors, allow uncontrolled spontaneous differentiation. They form all the tissues of the embryo, and they form sperm. So they're totipotent stem cells. They'll form everything. So you have a pluripotent embryonic stem cell and a totipotent embryonic stem cell. Okay? Now, just out of curiosity, yeah, go ahead.
I just noticed that the inhibitor that's used for this one, you're only using rats and mice, and for the previous one with the stem cells that you retrieve from the blastomere,
Yeah,
you use humans.
exactly.
So
We're not quite out to they're not quite out to humans yet with the blastomere stage. These guys right here.
Mm-hmm.
They haven't quite done humans yet, but they're getting there, especially the Japanese. They're real ahead. They're ahead of everybody else in the field. And the reason the well, let's see how to put this. In the Japanese culture, they do not transplant. They do not give away body parts. It's part of their religious culture that when you get buried, everything gets buried with you. So you don't transplant livers or parts or anything. You don't give anything away. But it's okay if you take things from others, especially others that aren't together to begin with. Okay? Basically, the way to put it. And anyway, so we have embryonic stem cells derived from either blastomeres or the inner cell mass. If it's blastomeres, we can form sperm, either one. Now, we have some problems here. They will spontaneously differentiate if you take the inhibitors away. In vitro, they'll form what are called embryonic bodies, which are okay. But if you put them in a live organism, they will form a mass of tissue called a teratoma. And a teratoma is a cancer. That's not a good thing to have, okay? But they will form all the tissues shown here. And the other problem that they have is the fact that these things are called allogeneic, meaning if they took a blastomer from, say, one of my wife's eggs, and tried to transplant it into you, it wouldn't work because you would be what would be called an HLA mismatch. So it would be something would be rejected. Either you would be rejected or what was put in would be rejected. So there's some problems with embryonic stem cells. And the two major problems are they're allogeneic, like I said before, and they form teratomas. Now they've done some studies to get around these problems. And the first one is the allogeneic, which is mismatch. Donor cells are rejected by host as non-self, or it could be the other way around. The donor cells might reject the host. Depends on how much you transplant. And anyway, the idea is you have a donor egg with a donor nucleus, come down around here, and it forms donor antigens on the cell surface. And these are the ones that recognize self versus non-self. So you put this donor antigen, which is a diamond here, into another individual that has a half moon as an antibody, that's the recognition molecule. They don't match, so this cell is rejected. So it's called an HLA mismatch. Okay, now, the way they've gotten around that problem is a technique called therapeutic cloning. And in that technique, they take a cell from the recipient, take out its nucleus, and take out the nucleus from the donor egg and replace it with the nucleus from the host. Let it go through this process down here. So now the donor egg is producing host antigens. And the host antigen matches host antibody. It thinks itself, we're fine. It's not rejected. This process takes about a year to do, and it's very costly because you have to go through a lot of these different things to get ones that work. Yes?
Again, is that the same as a nuclear transfer?
Yes, that's exactly the same as nuclear transfer.
Okay.
That's how they make Dolly, actually, the sheep. Now, we also have formation of teratomas, which are cancer cells. And that's when naive embryonic stem cells are implanted inside an individual, and then they spontaneously differentiate. And again, that is a problem. So the way they get around that is, what they do is first therapeutically clone, so you're going to have an identical match, grow up as many cells as they can, where they want, remove the inhibitor, form the embryonic bodies, allow uncontrolled spontaneous differentiation, form all the cell types you want, and then just select only the differentiated cell type for transplant. And this adds about another year to the process. It'll take you about 2 years to do embryonic stem cell with therapeutic cloning to get a transplant tissue for yourself. Plus 2 years of time plus a lot of money to do this sort of thing. But it does work. It does work. Yeah. Some additional problems they will encounter that they don't know about yet. One is they have to differentiate to the terminal cell type. So it's no longer unlimited differentiation potential. Because now you've got a progenitor cell or the differentiated cell type. So the cells are no longer plastic. The cells now have a limited lifespan. So if you go from one cell to where you are now, that's about 30 population doublings, or maybe 40. So maybe you're now at the cusp end of where you should be, so you can't live as long as you thought you could. Differentiated cells deal very poorly with existing tissues. Existing tissues really don't know what to do with already, skeletal muscle cell. If you try to implant it into another skeletal muscle, it really doesn't know what to do with it. It's like, okay, what am I supposed to do with this? I'll wall it off with scar tissue and just stick it off to the side somewhere. I'll accept naive cells, but I'm not going to accept something that's already differentiated. They would find that out if they read the literature from about I've been in here 35 years, about 40 years ago, which is where I started. Because we tried to transplant differentiated tissues into differentiated organisms, and it didn't work. It would form scar tissue. But most of the time, people don't read the back literature. They like to do things on their own, but they'll find out about it eventually. So it walls it off. And another thing they're going to find out, and they haven't found it out yet, and I'm just beginning to tell them, just to be nice about the whole thing, is it has to be gender specific. You cannot transplant male cells into females or females into males. And the reason is hormones. They work differently in cells. Yeah. And they haven't quite done that yet, but they'll find that out. Okay, now we go to my favorite lab, which are the adult stem cells. And like I said, I've been doing this for quite a while now. And I started out with, yes.
I'm sorry.
Go ahead.
Just before we get, so the very first slides that you were showing when you gave the definitions of cells, that could go for either embryonic and stem, or was that just for embryonic?
No, embryonic or adult, either way.
I mean, oh yeah, or adult. Sorry.
Yeah, it can go for either one. You can have totipotent, pluripotent, progenitor, germ-layer lineage for both. It's just general categories of how you categorize cells. Now, I name my cells according to how they do things. Other people in the adult stem cell world name them according to where they come from, which leads to kind of smoke and mirrors like, I've got a fat-derived stem cell. Well, or I've got a stem cell derived from fat. Well, that's nice, but what does it do, you know? Or I have one from muscle, or I have one from bone marrow, or I have one from this, or I have one from that. Great, but what does it do, you know? So I've kind of narrowed it down as to what the stem cells do, and I'll show you where you can get them in a minute. But anyway, yeah, same definitions for both. So I started out with progenitor cells. I had isolated two compounds when I was in graduate school. One compound would stimulate skeletal muscle formation. The other compound would inhibit scar tissue formation, which is really cute. And we needed a population of cells to work on, and in fact I used myoblasts, which were unipotent progenitor cells. And then we were isolating all these cells, and I think it was on September, that would make it Labor Day weekend. Standard procedure is to freeze the cells in liquid nitrogen, store them, and then come back, and that's what you do for differentiated cells and progenitor cells. We had a progenitor cell. So we had just isolated 60 million cells, and we were ready to freeze them down, and we looked in the lab, and there was no liquid nitrogen. So we looked around the whole medical school, and there was no liquid nitrogen. And, you know, so we went to Air Gas, who's a supplier for Macon, and they shut down early for the weekend, so there was no liquid nitrogen in the whole town. So it's like, okay, draw straws, who gets to stay and feed the cells, or who gets to go off on vacation? Well, nobody wanted to stay and feed cells, including myself, because I had planned a 5-day vacation. So we did a Hail Mary experiment, which was we froze them down in a Revco at minus 80 degrees centigrade. We just stuffed them in there basically, shut the door, prayed, oh please God, and then came back 5 days later, took the cells out, plated them, and looked at them, and boy, those things don't look like progenitor cells. These myoblasts are rectangular, and what we came out with looked like little stars. It looked the same. Well, we tried our muscle morphogenetic protein to form muscle. But at the same time, we were testing factors for Amgen, and Sigma, and Genentech, and all the big, you know, factor companies. And we had factors to form cartilage and bone, and lo and behold, we put it on the myoblast, didn't do anything. We put it on this cell, it formed cartilage and bone. That's interesting. We had endothelial cell growth factor from Amgen, put it on our muscle cell, didn't do anything, put it on this cell, formed endothelial cells. We had EPO from another company that formed erythrocytes, supposed to form erythrocytes. Myoblasts wouldn't form anything on these cells, it formed erythrocytes. Okay, we didn't have a progenitor cell. What we had was a germ-layer lineage mesodermal stem cell. So serendipity, pure serendipity, nothing else. That's how we found that cell. We found the ELSCs basically the same way. Came up, I think this was more in the springtime, a few years later. And we grew up these cells, and we were ready to go off on vacation, you know, this sort of thing. And we had like 100 plates, and we had to trypsin release to get the cells off to freeze down in minus 80. And near the end of the day, nobody wanted to do this. And the one difference about the germ-layer lineage stem cells as opposed to progenitor cells is once progenitor cells or differentiated cells reach a condition called contact inhibition, they'll die. They just croak. dead away. Germinal lineage stem cells, they'll remain perfectly fine as long as you feed them. So we just tanked the cultures up full, stuck them in the incubator, knew they'd survive, came back after vacation, looked at the cells, and yes, we had a monolayer of cells, but we had all these little cells on top of them. You know, kind of what's going on here. So we separated the little cells from the bigger cells, and we tested them and all the factors we had tested before, and they did exactly the same thing, mesodermal lineage, but we also had hepatocyte growth factor at the time. We tried it on our germ-layer lineage mesodermal stem cells, didn't do anything. Tried it on these guys, they formed liver cells. That's an endodermal lineage marker, or endodermal lineage cell. We also had nerve growth factor in the lab, which we were testing on the cells, and it wouldn't do anything. We tested on these cells, and it formed nerves. Well, it's not a mesodermal marker, it's not a mesodermal line, it's an endoblastic cell line. And then again, we found blastomeric-like stem cells, again, by serendipity. Everybody says this stuff looks like cell trash because it's real small. And we thought it was cell trash and we kept throwing it away every time we passed it to cells. And we went on vacation, tanked it up, came back, and this thing was covered with all this little stuff. Now, except for my daughter's room, I know of no other place where trash multiplies, if you know what I mean. You know? So we think, well, okay, this might be a cell. So we separated them, and I put 1,000 cells in a big flask, left it in the incubator for 2 weeks, came back, and there were 1 billion cells in the flask. Well, okay, that's not trash. So we characterized it. We did karyotypic analysis on it. They had a normal karyotype, so these were real cells. And we tried, you know, all the growth factors we had, and it formed everything the pluripotents did. And we thought, well, maybe it's just a small pluripotent stem cell. We also have an antibody in the laboratory that will recognize sperm. And we've always tried it on the pluripotents to see if they were, you know. So we tried it on this cell population. Lo and behold, side by side, the pluripotents formed everything pluripotent. These guys formed everything pluripotent, and they lit for the antibody that formed sperm. The other ones did not. So we had a totipotent stem cell. So serendipity is how we found them, but we followed up on what we found, and I'll show you some cutesy results we've been getting with these.